Edison TV. I'm Rachel Carroll, here today with Medlab, a unique company whose novel phase 3 drug and proprietary delivery platform aims to harness the power of cannabinoids in order to address chronic pain. Our analyst initiated this week valuing Medlab at 201 million Australian dollars, or 59 cents a share. That's a 250% premium to where the shares are trading today. I'm here with Medlab's Chief Executive Officer, Dr. Shawn Hall, to talk about how he plans to close that gap. Shawn, delighted to have you with us here today.
Rachel, thank you very much for your time.
So you've got a really interesting background, Shawn. Perhaps if you could start with a little bit of information about yourself, how you came to be CEO of the business, and then introduce us to Medlab and its programs.
Yeah, a good question. And like a lot of people that have ventured into healthcare, it's more one of personal effect than anything else. My family has a history of being investment angels or in rescuing healthcare businesses, building them up, and subsequently, exiting at the appropriate time. One of the last family operations we did was a company called Biotechnology. And it became relatively quickly Australia's leading vitamin and mineral supplier to doctors. So the difference is you wouldn't just walk into a pharmacy and buy it. A doctor in a consultation would script it like they scripted an orthodox medicine. And part of the rationale for that is we saw no difference between a vitamin and a pharmaceutical agent if it inferred a health benefit. Off the back of that, we had a small biotech where we were playing with meloxicams, which is an analgesic, and we were making them effervescent so that people in the field, people in remote areas could place this on the tongue. The effervescent activity would work, and they would have a non-opioid analgesic, but a temporary relief of pain. So think effervescent acetaminophen. Blackmores came along, which is one of Australia's leading vitamin suppliers in pharmacy, and bought us. But pre all that, my brother passed away with a cancer that everything I learned about oncology proved to be absolutely useless when it came to giving him quality of life or extending that life. So Alex really was the catalyst for me to go back into research and to look for a different way to treat patients that are suffering the gamut that we call oncology, that is beyond just the cancer itself. And, in that, when Blackmores came to purchase Biotechnology, I saw an exit. I saw the ability to pivot back into something that I know, I love, and a real fundamental ethical belief that I could actually do something of benefit and really put some emotional and knowledgeable clout into building a drug that we firmly believe is going to greatly impact human life moving forward. So here we are today. I started Medlab. I self-funded the first 6 million Australian dollars. We built our own genetic laboratories in Australia. We're one of the few that actually own federally regulated genetic laboratory outright. And we started looking at several core areas, all in the area of chronic diseases. It's great for pharma to turn around and go, I'm going to do a blockbuster and we're going to have a new diabetic type 1 drug. But what about all those other people in those peripherals, those mild to moderate areas that don't need the blockbusters? And this is where Medlab clicked in. We got interested in cannabis because of the fact that I was following GW Pharma early in the day. I actually was looking at them for medical relief for Alex during his final days. And I thought I have the ability to improve this. They were on the right track. And there was real legs in the fact that an equimolar of CBD and THC could provide a non-opioid analgesic type response without the side effects. Hence, we have the NanaBis program today. The question is how do we make it bioavailable? The problem with most drugs that we take today is they're just not soluble. The most convenient form of pharmaceutical medication is for you and I to swallow it. But from a drug capability point of view, to maximize the response of that drug substance, to absorb the most of it in the body, if I can just be very motherhood in how I put this together, swallowing a tablet is the most inconvenient way to take a medicine. And you can see that if you follow the course of medicines, typically what we do is we make them stronger. We make them stronger, we make them stronger. And the statins are probably the best example I can give of that. So I went back to an old mentor, a friend, and I brought him into Medlab. His name is Dr. David Rotolo. He's based in Southern California. And many people still remember Dave from his work some 4 or 5 decades ago. He was the founding principle and the original patent holder for micellization. And the whole topic of that was how do you take something that is relatively insoluble, so let's call it fat soluble, it's not absorbable, and you make it quite the contrary. And we built on the old micellization technology, and we came out with a delivery platform that's rather unique, that has been validated. As we have seen recently, it's getting world patents being granted. I think the US is the only region left that has not made final determination. And we use the NanoCelle delivery platform to enable or enhance the delivery of medicines in the case of NanaBis, which is our CBD THC non-opioid analgesic, to allow lesser dosing of the drug substance to reduce that overall side effect burden, whilst at the same time allowing us to really trigger that efficacy that we're seeing in advanced cancers and in many cases with intractable pain today. I must apologize, sorry about that. My puppy is in the office at home, and he thinks it's breakfast time. So,
no problem. It's a tremendous and deeply personal backstory. Thank you so much for sharing.
Pleasure.
I want to touch upon. So, you recently announced a shift to a synthetic formulation of your lead candidate, which is hopefully entering phase 3 trials imminently. Could you talk a little bit about the rationale behind this decision and how, if at all, it will impact phase 3?
There's a few lessons to learn in this. Botanicals are great, and I can understand why the average person is pro botanicals, especially now when we talk about whole life and whole plant and stuff like that. But the reality is if we're going to make a drug substance, then we need to bring a precision and a quality into that medicine. Because to be a drug substance, you have to be approved, in your case, by the FDA. In my case, our equivalent is the TGA. And we got our botanical substances to 97% purity. But the amount of work we had to do pre-manufacture was horrendous. It's not that we didn't want to do it, but it drives up the cost of the product. And it really starts to dictate what crude material you can use and you can't use. The amount of adulterated crude material out there on the global market is, I've got to be honest, it's disgusting. And we have looked at several hundred different samples. So in speaking to the FDA during our pre-IND meetings, we were talking about the role botanical can play versus synthetic, and it came relatively clear that they obviously have a more akin appreciation to synthetics because it's biosynthesized, you can grow it in a lab, it's standardized, and you just get precision at every step. Our problem was the technology for synthetic CBD, so a 100% pure dronabinol, just didn't exist. And we kept working on the botanicals, and we were aiming, and still are now, for a 98.2 or 0.3% purity index. Now, that little 1% in organic chemistry worlds is like us today going, we want to fly to Mars. It's big. So in January of this year, working with a partner, East Coast Space, they actually made for the first time, and they were able to categorize it, a 100% pure dronabinol, which met all our starting requirements for a drug substance for NanaBis. So we made the pivot. So today here we have a synthetic 100% CBD that has drug master file recognition with the FDA. We have completed manufacture of a 100% synthetic dronabinol or THC by common name. And we are now working on application for drug master file status, again, with the agency. The pivot allows us to do several things that the botanical couldn't. We're eliminating multiple stages in the manufacturing cycle, which speaks to cost speaks to time, and ultimately speaks to patient price. The other thing that we're getting is the yields out of the synthetics, because they have pretty much a zero margin on waste, is 3 to 4 times that of the same volume of the botanical. So as far as NanaBis is concerned, from a regulatory point of view, we believe we have bolstered and we have de-risked considerably the CMC package that goes to drug registration. We have brought a higher degree of precision into the drug substance. We have significantly been able to reduce the cost of manufacture and the time to manufacture. If you think about a plant for a second, you have to grow, harvest, extract, and then manufacture. To get a full adult plant in the marijuana index, you're looking at some 16 weeks. But right here, right now, we can turn a lab on, we can produce the synthetic, and at this point, the synthetic has a 100% plant identification to the isomer. So I think we're sitting very strong. And when we speak about synthetics, we're speaking it in line with drug registration models. The company is still pursuing botanicals. It's still pursuing moving from that 97% to that higher 98.2%. But at this point, moving forward with NanaBis, it will be a synthetic play. We've brought that in to de-risk the overall application that we ultimately will be putting to the FDA for registration.
That's incredible. Thank you so much, Shawn. And I'd like to go back, if I may, to the, you were talking about the delivery platform, which is your proprietary platform, NanoCelle. So this to me seems unique in terms of drug delivery. Could you talk us a little bit through how it actually works, why it's more efficient, and what other potential drugs it could be used for?
Yeah, actually, great question. One of the things that we're exploring now, we have, let me start by saying we have some 15 odd partnering conversations underway, and 90% of them would be Northern Hemisphere. A couple of those are larger companies that traditionally have a large generic portfolio in global territories, and that leads to 50-60% of their global revenues. So we're getting a lot of questions from potential partners and also research associations on being able to diversify the application of NanoCelle. Here and now, in the medicines that we've delivered, and three are Australian listed medicines under TGA, we favour the buccal membrane, so the interior of the cheek. And the reason for that is we can cross it, we can get the drug substance released in the body, and it can get systemic very quick. We've had interest from medical companies that look at medical lotions on putting it onto the dermis, and we've done a lot of development work in that says without a doubt there is no problem. We've got academic write-ups on how to convert this to a nasal spray, but in our partnering conversations, they're really going one of two ways. We have those companies that have those generic profiles that want to re-enter branded lines and want to put some new marketing fodder on their generics. So can we improve a generic, whether it is buccal or whether it is nasal or something else? The answer 100% is yes. And we've done at our Sydney labs now some 23 generics across 5 or 6 categories of medicines where we have systematically improved it. A lot of the work we originally did was, believe it or not, in statins. Because the atorvastatin molecule was it's a difficult one to play with at the best of times. Notwithstanding, the other area of interest, and we're starting to field a lot more conversations, both from academia and also from business partnering, is can we use NanoCelle to deliver an immunotherapy or a vaccine by the oral buccal membrane? Can we effectively, quote unquote, and some of my fellow doctors will shoot me for saying this because it's to motherhood, but can we remove the needle from the equation? Theoretically, the answer is yes. So we are going to start working on a couple of substances. One broad stroke is protease inhibitors, whereby we're looking at being able to nanocell it so that we get nanoparticle structure in the drug substance, and then look at being able to deliver that payload that payload to then become efficacious, and a lot of that will rely around other development work we need to do along the lines of drug dosing models. So from an academic point, 100%, we all know that academia to real world, there is a vast chasm there, and we've got to cross that bridge. But we're feeling very confident about it now. How does it work? In simplest terms is going back to Dave's original work in looking at mycelials. We took a mycel structure and we standardized it. The problem with mycelials today is you could put 30 different products that are all mycel on a table, and they will all look, smell, taste, and perform differently. In fact, most probably are not mycelials. So we've developed a characterization structure that puts the finished product at a certain range of size that's well within the nanoparticle scope. We have built a trigger to release the drug substance. So usually we emphasize pH as the environment to release the drug substance from the environment. The scaffolding of the delivery platform falls away in plasma and is excreted. We have not seen in our 5 years of working with NanoCelle a toxicity buildup within the body or autoimmune response that you've typically seen in other delivery platforms. And I'm comfortable in saying this because to date, 5 years that we've been looking at and working with NanoCelle, we have put out, I think last count was about 300,000 NanoCelle drug substances in various markets, whether it's under a compassionate use program, whether it is an approved substance. As I said, we've got 3 or 4 that are listed medicines on the TGA here in Australia, or in clinical trial work. And in many cases, we track the patients for a long period of time. And we've got one program now where for NanaBis, we're tracking, I think we're at around about 911 patients. Don't quote me literally on that. And a bulk of them have passed the 6-month mark. A number of them have passed the 12-month mark. And one of the things we're looking for is this side effect profile. Now, typically, if we look at other delivery platforms, and one in particular that comes to mind was one called liposomes, we would see an autoimmune response called CARPA happen around about 8 to 12 months. These are things we're not seeing when it comes to NanoCelle. So we do have something unique. We have something that's very easy to manufacture, and we have something that is diverse.
That's really exciting. Thank you so much. And so, Shawn, you're obviously based in Sydney, but it's been great to see you in the United States presenting at investment banking conferences. Can you talk a little bit more about your US strategy in particular, and what investors should look out for to monitor your progress over the next 6 to 12 months?
Yeah, for sure. First, I wish I could be there in person. This COVID has sort of put a dampener on all that. So I suppose these virtual means are going to be the way of life for a bit longer. It's clear that the US is the biotech hub of the world. I mean, let's be honest. And we believe we have something that is unique, something that is disruptive, something that can really impact lives. So to that, we feel we need to be part of the US community. Notwithstanding, when we look at NanaBis, it is an FDA play. Part of the reason it is an FDA play is your opioid crisis not only has been well publicized that the general man on the street understands it, but it is also the largest market. So if we're going to affect change, if we're going to do something worthwhile for people, it makes sense that the US is where we want to go. Looking at our US plans, I'd love to think going forward, and not speaking out of school here, that there is a role Medlab can play with the US capital markets. I think as we've bolstered down and we've worked on our assets and we've valued those assets and we've got protection around those assets, we need to expand from Australia. At the end of the day, we're a global marketplace and our drugs are going to end up globally. So let's move to the US and work with the people there.
And there we have it. So Shawn, it's been great talking to you. Thank you so much for coming on Edison TV.
Thank you very much for having me. It's been a real pleasure. I hope I get to meet a lot more of the US side people in the near future. Best case, I look forward to returning to the US and being able to meet face to face. Thank you.
Fantastic. And if you'd like to learn more about Medlab and the investment potential for the shares, Please do read Edison's July initiation report, which is freely available on the Edison website, or contact me personally at rachel.carroll@edisongroup.com. Thank you.